Hello, everyone, and welcome to today's webinar, the MADx Multiplex Universe. My name is Amy Wilson. I'm a Senior Product Manager here at ALEX² and your moderator for today. ALEX² is pleased to be the exclusive distributor of the macroarray diagnostics multiplex IgE testing platform in North America. I'm also pleased to have with us today Dr. Martina Aumayr. Dr. Aumayr is the Head of Research and Development at macroarray diagnostics. Today, she will be discussing the ALEX² multiplex IgE platform, including the allergen array cartridges, manual and automated instrumentation, and analysis and interpretation software. Before we get started, we just have a few housekeeping items. First, today's presentation is being recorded. You will receive an email within 2 to 3 days with a link to access the recording that can be shared with colleagues who are unable to attend. Second, we'd love to hear from you during today's presentation. Please submit any questions you have through the chat box. If we can't get to your question during the Q&A following the presentation, we'll be sure to follow up shortly after. Finally, there will be a brief survey at the end of the webinar. We appreciate any feedback. And now I'd like to welcome today's speaker, Dr. Martina Aumayr.
Hello, nice to be here. Thank you very much for the nice introduction. So today I will introduce you to the MADx Multiplex universe and show you all the products it includes. Good. So in the beginning, I will briefly summarize the basics of molecular allergology. So just to show you some definitions, my main focus will be about our ALEX technology. This is our allergy multiplex diagnostic test. So about the principle, why we combine extracts and molecular allergens, what CCDs are and why it is important to block CCDs, and about unique molecular allergens on the allergy test. The allergy test can be performed manually and with fully automated equipment, and I will show you both workflow options. Also, the results can be analyzed with the RAPTOR server software, and I will give you an overview about what the possibilities with this software are. So let's start by having a look at the basics of molecular allergology. So what is the principle of molecular allergology? You have the allergen source, and this is usually the allergen producer. So for example, a house dust mite, and an allergen extract is a mixture of allergenic and non-allergenic proteins. A molecular allergen is a single pure protein that is capable of inducing a histamine release. And here you can see another good example of the difference between extract and molecular allergen. So let's take, for example, the egg. And as everybody knows, you can separate the egg into the egg white and into the egg yolk, and it looks different. But also on a protein SDS page shell, those extracts look different in their allergen composition and also in the concentration. And you can further purify one single protein, in this case the GALDI4, and this is the pure molecular allergen. So why molecular allergens instead of extracts? So extracts can be contaminated with other antigens, and there are many publications that show that extracts, there are several references here, that extracts are contaminated with other allergens, leading to false positive IgE results. I would also like to show you a picture of commercial allergens. Here, a picture of commercial house dust mite allergens that can be bought from different companies, and they vary greatly in their allergen composition. As you can see, they all look completely different. And the major allergens, the DERP1 and the DERP2, are available in different concentrations, and leading to inaccurate diagnosis. And essential major or marker allergens like the DERP5 or the DERP23 are sometimes underrepresented or not even present in those extracts, leading to false negative results. And of course, the cross-reactivities are difficult to detect. IgE results on molecular allergens give more detailed information on the actual sensitization status. So the marker molecules help in assessing the risk for clinical manifestations and determine if anaphylaxis might be imminent. You receive a complete resolution of cross-reactivities, and an appropriate immunotherapy can be chosen more easily. Also, the patient has an advantage as different dietary recommendations can be given. So, for example, the patient can be told if the baked apple will be tolerated or the fresh apple won't. Molecular allergens can be purified from a natural source, and this means that the raw materials, such as the peanuts, for example, they can be purchased from the supermarket, can be defatted, extracted with different buffering systems, and chromatographically purified according to different properties into one single protein. Another way is to produce the molecular allergens recombinantly in microorganisms, and this is done by introducing the gene of the desired allergen into bacteria, for example E. coli, allowing them to produce the proteins and harvest them, and again purifying the protein chromatographically on the basis of the properties. Both options have advantages and disadvantages. So the natural allergens, they're really easy to produce. You simply go to the supermarket, buy the nuts, and then purify it. You have all the isoforms present. And sometimes it's important to have all the isoforms for the allergy diagnostics. However, some allergens might not be present in high quantities. So this means that you might need kilos of peanuts to purify one single protein. You have to be very careful that no other contamination with other allergens from peanuts will be there, and natural molecules can contain CCDs. Recombinantly expressed proteins have the advantage that the quality is always the same and does not depend on the starting material. It is always an exact replica of the protein, and you don't have any CCD on the recombinant allergens. However, you only have one isoform because you only introduce one gene of interest. And if you would like to have several isoforms, you need to express more than one protein. And the expression is not always feasible, since some of the proteins are toxic for the bacteria and they are not allowing you to express it. So now let's have a look at our products. And here you see an overview of our products, hardware and software. We have both an multiplex IgE test for the diagnosis of allergies. It is called ALEX or ImageXplorer, as well as an IgG multiplex test that helps analyzing and monitoring dietary behavior. And this is called FOX or Food Explorer. Our hardware includes at the moment the manual imaging device, the ImageXplorer, and our fully automated system, the Max 45K. And it will soon get a small brother the miniature automated system, the Max 9K, which will be available in September for smaller sample numbers. The RAPTOR server software is the software that connects both the products and the hardware together. So it's usable for all our multiplex tests and all our hardwares. And the Raven interpretation software helps interpreting the result. So this is how the ALEX looks like, and you see it has a lot of parameters on the test. So what is the ALEX or ALEX²? Our product, the ALEX² is a solid phase immunoassay for the detection and quantification of specific Ig and total Ig in the blood. So the multiplex test offers the simultaneous detection of 296 test features and the largest panel of molecular allergens on the market. It combines extracts, 117 extracts, 117 molecular allergens and total Ig all in one test. And the balanced allergy panel recognizes both the needs for inhalative allergens such as pollen, food allergens, but also special diagnostic needs like insect venoms or latex. And here you can see an overview on the different allergens ranging from pollen, molds, mites, dander and epithelial, venoms, and plant-based and animal-derived food. So it has unique molecular allergens, so more than 50 of the allergens on the ALEX test are unique, so they're not found with any other test system. And it is the only CE marked Ig test with an integrated CCD inhibition function. And it therefore provides a comprehensive view of the patient sensitization status and empowers the physician to quickly and precisely prescribe the fitting therapy. So what is the ALEX technology? So the core technology is based on a two-phase manufacturing process. And while usual or other multi-parameter ELISA assays use the identical settings for the immobilization of all antigens, we are able to adjust these conditions for each allergen individually until the optimal result is obtained. In a first step, the allergens are coupled to activated nanoparticles, and this allows for individual and combinatorial optimization of each antigen coupling process. So each antigen is attached, reflecting its biochemical properties and specific requirements for stability, which preserves the full epitope complexity of the source preparation. Also, the nanoparticles multiply the surface of the solid phase, presenting the antigen during the immunoassay and thereby enabling highly sensitive detection. In a second step, the antigen-bearing nanoparticles are deposited contact-free onto a solid phase matrix, forming a macroscopic array of individual assay parameters. So if one imagines now that one spot contains, so a spot is one of the dots on the arrays, if one spot contains only one molecular allergens and the other spot contains the whole extracts, you will understand that the sensitivity for molecular allergens and extracts might be different. So with extracts, the available surface has to be shared between different molecular allergens, and therefore only limited space of the surface is available for each of the allergens. In addition, some of the allergens are present only in minor content, and some are simply underrepresented. So, but for molecular allergens, the complete surface is available and therefore the sensitivity is higher as the available surface has not is not shared between with other molecular allergens. So a combination of extracts and molecular allergens has the advantage that you can use extracts where molecular allergens are not well characterized and redundant due to a high degree of cross-reactivity, and you can use molecular allergens where extracts are unstable. So what is the manual assay procedure, or also the assay procedure that is then performed in the automate automatically? So you prepare a small volume of serum or blood, and this is sufficient to detect the patient's sensitization status. So in the first step, the particle-bound allergens react with the specific Ig that is present in the patient's sample. After the incubation, unspecific Ig is washed off. The procedure continues by adding an enzyme labeled anti-human Ig detection antibody, which forms a complex with the particle-bound specific Ig. After a second washing step to wash away the unbound detection antibody, substrate is added, which is converted into an insoluble color precipitate by the antibody-bound enzyme. Finally, the enzyme reaction is stopped by adding a blocking reaction. The amount of precipitate is proportional to the concentration of specific Ig in the patient sample. The manual assay procedure or lab procedure is followed by the image acquisition using the ImageXplorer device, and also a small ImageXplorer device is built in the MAX 45K or MAX 9K. The quantification is a colorimetric readout, and the test results are analyzed with the RAPTOR server software and reported in Ig response units. One of the key features of the immunoassay is the blocking of clinically irrelevant CCD signals. But what are CCDs? So CCDs are cross-reactive carbohydrate determinants, and those are sugars located on allergens such as plants, insects, or mussels. And these CCDs on the proteins are able to induce the production of IgE. And in serum-based allergy diagnosis, antibodies directed against CCDs give the impression of a polysensitization. However, these anti-CCD IgEs do not seem to elicit any clinical symptoms, and diagnostic results caused by those CCDs are therefore regarded as falsely elevated or false positive. So usual test systems do not block the binding of the antibodies directed against CCDs. So if we have a look now and you have a serum without any specific IgEs, you get the true negative signal. If you have antibodies directed against the specific allergens, you get the true positive signal. If you have both IgEs that are directed against allergens but also against CCDs, you will receive a falsely elevated or higher signal. And if you have only IgE antibodies directed against CCDs, you will receive a false positive result. So how do you get rid of CCDs? So you could either use recombinant molecular allergens that are not glycosylated, and on ALEX² there are 178 molecular allergens, or you incubate the serum with a blocking inhibitor, and that's why a CCD inhibitor is added during the serum incubation step. So to emphasize this with an example of an ALEX array, here is one test performed without CCD inhibitor and with CCD inhibitor. So the same serum sample, And you see that without the CCD blocking, there are a lot of falsely elevated or false positive results. So about 20 to 25 of the allergics are positive to clinically irrelevant CCDs. And this is also an example from an Austrian clinic where a patient was diagnosed for peanuts. And you see that quite a lot of peanut allergens were elevated without the CCD specific antibody blocking. However, when a CCD blocking inhibitor was added, all the allergens, including the risk markers, were put to 0, indicating that the signal was only due to CCD specific antibodies, and therefore no emergency set or diet would be required or was required. So CCD epitopes have a major impact on IgE testing and can lead to false positive or falsely elevated results, especially extracts and purified components from plant-based food, venoms or mussels contain CCDs, whereas recombinantly produced allergens are CCD-free. The use of a CCD inhibitor is recommended and is essential for high specificity of the results. So the ALEX² key features, and we have already heard some of them, are as much information as possible with the largest panel of molecular allergens and extracts. We have unique allergens, which we will have a look in a minute, and there is a Raven interpretation software as well available. It is also, or it also tries to be as specific as possible. That's why we use a CCD inhibition during the serum incubation. And we also try to be as reproducible as possible. That's why we emphasize on recombinant molecular allergens to reduce the dependence on the quality of extract raw material. So ALEX² has more than 50 unique allergens that are not found in other test systems, and there is a big focus on nut allergens, legume allergens, seafood, furry animals, mites, but also special ones like cannabis or edible insects. And the ALEX² covers all important protein families. So what are protein families? Proteins or allergens are systemically categorized in families based on their amino acid structure, sequence and their structure. And the ALEX² contains storage proteins, polkacin, profilins, serumalbumins, tropomyosins, lipocalins, PR10, LTPs, parvalbumins, and many more. But why is it important to test so many allergens? And I would like to pick the example of the nut allergens, and the ALEX² contains a lot of nut and legume allergens. So in total 17 nut allergens plus 5 extracts and 11 legume allergens plus 4 extracts from various different nuts. And it is important to test those different allergens because first, they differ in their clinical relevance. So most of the allergens belong to the group with a high risk to develop anaphylaxis, and all those red molecular allergens are marker allergens for anaphylaxis. And second, they differ in the cross-reactive behavior. So some have a broad cross-reactivity with homologous allergen sources like the PR10. So we don't have so many allergens from those for the nuts, but others have a very limited cross-reactivity to homologous allergens from closely related sources. That's why it's important to test so many different storage proteins from different nuts. So why use a multiplex test and not a singleplex test? A singleplex test only shows part of the sensitization status. It is highly reliant on the patient information and the physician, and it sometimes needs more than one run to identify the correct allergy, especially in complex cases. A multiplex test shows or usually shows the whole picture, at least if the allergen panel is large enough. The anamnesis can be guided by the results, and usually this leads to a quicker route of therapy. Only one test run is usually necessary, but of course the interpretative burden is much higher for multiplex tests. So to sum up, the ALEX is a multiplex allergy test with the largest panel of molecular allergens on the market. It uses a combination of extracts, molecular allergens from allergen sources around the world, and total IG. 50 allergens are unique on the ALEX test, and it is the only test that offers automated blocking of CCD antibodies. It therefore shows a complete picture of the patient's sensitization status and helps doctors to diagnose the patient and prescribe the best and individualized treatment. Now we go to our hardware or the processing equipment. So here you can see our 3 imaging and processing devices. So the manual imaging device, the ImageXplorer, the fully automated multi-array explorer, MAX 45K, and the soon available small automate, the MAX 9K. And in our starting package, we provide all the necessary lab equipment to perform the manual immunoassays, so the shaker and the incubation chamber, the ImageXplorer, the array holder, and of course the software. And we have gone through the manual assay procedure before, but just quickly again, you prepare the serum, then you have a 2 hours incubation step with the samples, you have some washing steps, then you have an antibody incubation step for 30 minutes, you have again a washing step, and you add the substrate, which you stop after 8 minutes. Then you let it dry and analyze it with the ImageXplorer. So the ImageXplorer is the imager device for the manual analysis, and it captures one image of the array at a time, and it is integrated in our RAPTOR server software. It only requires a PC with USB port. And for the manual test, 100 microliters of sample volume is needed. The MAX 45K is a benchtop batch analyzer for fully automated processing and image acquisition. It can be used for the ALEX and also for the other multiplex test. It is fully integrated with the RAPTOR server, and up to 50 samples per run can be analyzed. Since one run is about 4 hours, you have a maximum of 150, or you can analyze a maximum of 150 samples per day. It has about 100 kilograms, and in Europe, it's classified as IVD. The workflow is in principle quite simple. You just prepare the serum, put it in the automate together with the reagents, and the rest is done automatically. I would like to show a short video of how the MAX 45K performs. So in principle, you have 2 rotors. In one you put the reagents and all the samples. And then the other one, you put all the ALEX arrays. Yes, so this is how it performs. Yeah, and soon in September 2022, our small benchtop batch analyzers for fully automated processing will be available again for all the products. It again will be integrated into the RAPTOR server structure. However, it will be a plug-and-play instrument, so no installation procedure will be necessary. And it can analyze 10 samples per run, and it only contains 50 kilos, so, and is also from the size much smaller and perfect for small or medium-sized analytical structures. Last, I would like to show you our RAPTOR server software. The RAPTOR server software is a cloud-based analysis hub for all the MADx systems, and the main advantage is that you have 24-hour access to the test results. You have an intuitive user interface for efficient workflow, the ability to use multiplex on demand, and I will show you in a minute what this means. An integrated way to interpret the results. Of course, since it's online, you have inbuilt online support function, automated software updates, so no need to install anything. You can customize your lab report, and there are several lab report languages available. Of course, you have increased data security since the RAPTOR server software is automatically backup and it is GDPR compliant. Again, I have a short video of how easily accessible the RAPTOR server software is and how you generate one picture. So in principle, you just open your Google Chrome web browser, type in raptorserver.com. You can log in via Office 365 or username and password. And you log in. There is a two-factor authentication is available. And then once you're logged in, you can say start measurement. The ImageXplorer that is connected will read the array. And you have the patient information on the left, the array picture on the right side, and the list of allergens on the bottom. You can sort those allergens as you like. And once you're done, you can save and approve the measurements. It goes to the approved measurements folder where you cannot change much, but you can make a preview of the report, and you can very simply include the interpretation, change the language, gender, age, and environment, and request the interpretation. You can download then the PDF report. And on the first page, you have an overview with the summary of all the allergens. Then you have some detailed information about all the detailed sensitizations and information about cross-reactive allergens, number of tested allergen sources, and then the Raven interpretation. So again, this is how it looks like: patient information and summary, detailed, and then the overview page. It is possible to customize the ALEX lab report, and you can, of course, change the color of the report to any color you would like. You can change the logo of the report and include your lab logo. You can choose which dates, patient, and doctor information are displayed on the report. It is possible to change the allergen sorting order depending on the values or on the alphabetically, include the cross-reactive information or not, add the interpretation, and many more other features. So it's really very flexible in customizing the report. It is also possible to display only certain allergens, and in some cases, too many results make it difficult to interpret the results. So doctors can choose from 15 predefined panels. So here you see an ALEX measurement with all the results, and here you see an ALEX results where only the pollen allergens are shown. So 15 panels are available and can be easily selected in the RAPTOR server. Sorry. And this means to avoid paralysis by analysis, so either choose from 15 predefined panels or generate your own ALEX allergen software panel. The Raven interpretation is an optional tool integrated into the ALEX² report software that allows allergen-specific comments to be displayed. It was developed together with clinical allergologists and researchers, and the comments per allergen source include a short description of symptoms, specific information on molecular allergens, and, if you wish, optional therapeutic suggestions. You can also include information on cross-reactive allergen families, and the Raven is therefore designed to avoid time, to save time by avoiding the need for extensive allergen database searches. Here are some of my references, and thank you for your attention. If you have any questions, please feel free to ask.
Thank you, Martina. This detailed explanation of the MADx platform has been very helpful. There are a few questions that have come in. Please feel free to enter any additional questions into the chat box. We'll keep an eye on that. So the first question is, what volume of sample is required to run the panel?
Can you please repeat it again, because I'm hearing you very low volume.
What volume of sample is required to run ALEX?
Okay, so the volume for the manual analysis is 100 microliter of serum. For the automated system, depending if you want to pre-dilute it or just put it in the automate, it's between 120 and 200 microliters of serum.
Okay, and is serum the only sample type they can use, or can they also test plasma samples?
You can also test plasma samples, yes. Not EDTA samples, but citrate and heparin samples can be used as well.
Okay, one more question. So is there a direct correlation between the level of specific IgE antibody measured and either the likelihood of allergy or the severity of an allergic response?
Well, this is a good question. So it doesn't necessarily have to be, so not the higher the value, the higher the reaction, the clinical reaction is. So sometimes it also depends on other factors for clinical reaction to happen. I mean, in theory, it might be possible that the higher the value is, the higher the reaction, but this is not always the case.
Okay, and I have a question for clarification on heparin plasma. They're asking if they could use lithium plasma or, I'm sorry, lithium heparin or sodium heparin.
To be honest, they can use both. I think so. There is no difference between those two.
Okay, great. And then we did have another question about how many of the molecular allergens are actually recombinant, but I know I've done the count recently here, because in the US we test all 300 allergens, and actually 144 of them are recombinant molecular allergens, so a really high percentage.
Yes. So I don't have the exact number, but I would say like nearly 150 allergens.
Yep. Okay. And okay, so a question is, someone's asking if they're running the ALEX panel or the FOX panel at their institution, what is the recommended quality control material to be used?
So we recommend to use the Lymphocheck from Bio-Rad SIG control. So here we also give reference values for certain allergens, but of course you could also use serum samples that you have in high quantities to always run with your test systems or with your runs.
Okay, great, thank you. Okay, one last question that's come in. Okay, I think they're asking here if this is CLIA exempt, but this is something I know you're not familiar with the US regulation, so we'll look into that and reply after the webinar.
Okay.
So thank you so much for your time, Martina, and thank you to our attendees for joining for today. We know your time is valuable and hope you found this seminar to be very worthwhile. We do have 3 short survey questions that will appear once the webinar closes. We would appreciate just a few moments of your time. Your feedback is invaluable. Thank you again. Please feel free to reach out with any additional questions.